For the Chevron SURE program, we investigated context-dependent variation in the legume-rhizobium mutualism across plant life history. This research was performed by Jean-Louis Ancheta, Justin Chavez, David Ramirez, Claire Savage, Christine Sorensen, and Dr. Kane Keller. Our research this summer focused on symbiotic species interactions. Symbioses are close interactions between individuals of different species. If it is a mutually beneficial interaction, then the two populations increase each other's sizes. These are referred to as mutualisms, and examples include protection, resource, dispersal, and pollination mutualisms. Our research revolves around resource mutualisms. These are those that trade nutrients for carbohydrates. Examples include mycorrhiza fungi and rhizobia bacteria, which you will be hearing much more about. Rhizobia are bacteria that form associations with plants, exchanging nitrogen for carbohydrates. These bacteria are capable of nitrogen fixation. Nitrogen fixation is the chemical transformation of atmospheric N2 to NH3. The air that we are all breathing right now is 78% nitrogen. While this is inaccessible to plants and animals, they are a group of microbes capable of converting this into a usable form. This is especially important to ecosystems, since nitrogen is often one of the most limiting resources in environments. However, a rose is not always a rose. Symbioses shift along a continuum from mutualism to parasitism. When the benefits of a symbiosis exceeds the costs, it's a mutualism, but when the costs exceed the benefits, it's a parasitism. Whether it is beneficial or not may often depend on abiotic and biotic aspects of the environment. For example, as nitrogen availability increases, the need for rhizobia may decrease, creating cheaters that use carbohydrates produced by the legume without nitrogen exchange. We may expect this in heavily fertilized soil.
For our study, we explored context-dependent variation in the legume-rhizobium mutualism over time. In particular, we asked, how does the fitness benefit the leguminous plants received from rhizobia shift over time during the lifespan of the plant? And how do these effects vary across a nitrogen gradient? To conduct this experiment, we ran a 2x4 full factorial design repeated across 10 time points and replicated 4 times per treatment for a total of 320 pots. To set this up, we sterilized soil, then either inoculated plants with rhizobia bacteria from a liquid culture or not inoculated. Then plants were fertilized weekly with one of 4 nitrogen treatments, either no nitrogen, a low level, mid-level, or high level. Plants were harvested weekly for 10 weeks. At harvest, we collected data on height, leaf number, branch number, nodule number, chlorophyll content, above and below ground biomass.
First, let's look at how height varied across treatments over time. Here, we have pink bars representing our no rhizobia treatments and green bars representing our rhizobia inoculated treatments, with the 4 nitrogen treatments along our x-axis separated by week. On our y-axis, we have height measured in centimeters. However, interestingly, we see that inoculation helped plants grow taller faster when nitrogen is more limited, whereas when nitrogen is less limited, uninoculated plants are growing just as tall as inoculated plants. Later in life, we can see that height plateaus regardless of inoculation status or nitrogen availability, indicating that plants are likely reaching a maximum height. When nitrogen is more limiting, there is a lag in that response, but they eventually catch up. This may lead us to wonder whether the treatments even worked. However, height is just part of the story. When we look at leaf data, we see similar trends early in the plant lifespan. However, uninoculated plants don't tend to catch up in leaf number to the inoculated plants across most levels of nitrogen availability. Therefore, while the plants are reaching similar heights, we see upwards of 4-fold differences in the number of leaves on the stems. So summing this up, the plants are similar in height, but there is much more growth on inoculated plants. Our rhizobia and nitrogen treatments certainly affected the plants, and interestingly, we found that fertilization still increased plant growth on inoculated plants, even at the highest levels of fertilization. We are currently processing our biomass data, which will likely elucidate these patterns more fully.
Conclusions and future directions. What does all of this mean? The effects of rhizobia take a short while to manifest. but then persist throughout the life. Initial smaller gains and reallocation of fixed carbon from leaf growth to rhizobia results in longer-term gains. Even when the mutualism is well established, the plants continue to benefit from increased nitrogen fertilizer. Although rhizobia provides a benefit, the plant can still utilize additional sources of nitrogen. Height tends to plateau and converge regardless of inoculation status. However, leaf data indicates substantial growth still occurring across treatments. We are still in process of additional data collection, such as nodule data and above and below ground biomass. Lastly, we plan to repeat this experiment with a broader nitrogen gradient to assess nitrogen saturation. Thank you all for your time and support. We would especially like to thank the amazing funders of our SURE programs such as Chevron, the Department of Education, CSUB, and all of the awesome support that we have received from the individuals listed here.